There we go. I think that does it. Hi, everybody. After a quite a long break, welcome to the Roach Crossing Weekly General Live Stream, March 24th, 2022, aka the big giveaway stream today, after being gone for 2 weeks. I really can't believe it's been 2 weeks. I'm just going to crack open this bad boy. Hello, familiar faces. Good to see everybody. Hope everybody's been doing well these last 2 weeks. Hi. Again, everybody. We got 4 people off the bat, and let's hope that by 8 we get a whole bunch more, because that'd be really cool after 2 weeks of not streaming. So I am joined by, and let me just go ahead and say hello to everybody individually. Hello, Jay, Joshua, Hylas. I'm just gonna call you Hylas from now on. Every stream I can't figure out if I want to say Regilla or Regia or whatever, so we'll just go with Hylas. Hello, Madison and Evan G. I am joined by Victor. I decided to just turn off the manted light for the stream. I don't know why. I never exactly thought of doing that before. Maybe I did, but it made the room darker. I guess also we've had the daylight savings time crap happen, so now it's would have been 6 p.m. I am so tired. Hello, Vocabulous. But yeah, and the day hours are increasing too, so there's that. It's been 2 weeks of daylight hours increasing, and thus it is actually daylight outside still. And we will be getting to the point where I will probably have to start pulling down the shade for doing the live stream, if not moving outside at some point once the yard gets cut back. I was supposed to do it tomorrow, but tomorrow's kind of a semi-big-ish day with things that need to be done, and thus I will probably get to it this weekend at some point or next week. The weather's still going to be kind of bad, so I've been looking forward to this. Going listen. Oh, foster kittens! That's really cute. So yeah, I am joined by Victor, who, in order to sort of satiate his desire to pester me during some of the past live streams, I took him on a very long stroll earlier, and so he should be satiated for some time. Yeah, I would love to start talking about the trip. I can't believe hello exotic wilderness. I can't believe it came and gone, and it's a blur. It's kind of like last year's Florida trip. Surprisingly, last year's Arizona trip wasn't really a blur. It despite being a blitzkrieg, a lot of some of the highlights really stick with me still. But last year's Florida trip was definitely a blur, and that's what this Alabama trip felt like. Granted, we were only there for 3 days. It did take a whole day to get down there and then a whole day to get back and whatnot. So, but I can't believe it's been that much time. We found so much stuff. All 4 of the things I went down there specifically for, we found in fair to great numbers, so I'm happy about that. There were some last-minute targets that we kind of slapped onto things that were 50-50 for those. I'll get into that later. A lot of good times. It was great seeing Alan and Satchel in person again, and going out and letting the memes and whatnot flow. And yeah, it was also very nice to get a taste of spring before it happens up here, because things are just barely waking up. I mean, I got some shoots of stuff coming out of the ground. The asters by my front door are just sort of starting to poke out of the rocks. So that's a pretty good sign. I have some, as far as native stuff, crocuses are starting to bloom, which is also why there's kind of a fire under my butt to get my yard work done, is because of all my spring stuff. But the downside is, if I go out in the front and clean up all the leaves in the areas where there aren't supposed to be leaves, I have I have one more ornamental bed that I get the leaves out of. I probably will. It's been rocks. It's been rocks since I moved in because that's what I liked at the time. But over as the days go by, it's so much work to maintain such a small area with just decorative rock. And so I will probably be mulching it and letting a ground cover of strawberries or probably violets move in between stuff, so I don't have to deal with pulling unwanted volunteers of some things. I mean, I love it because I get free plants that way, but I don't want common milkweed all over the place. I really don't want more. I mean, it's hard for me to sit and think of things that I don't want out there because I love all of my native plants. But there's things that I would like to seed less in certain places, and it's too much work with the rocks. So to mulch it shall go. So this week will be a giveaway. I'm debating how many giveaways I want to do in the stream, but there is a big one that I've had in mind for a while. So if anybody has friends or associates who are looking for some really cool bugs and would like to get them for free, it would behoove them to join in for this live stream and to stay perhaps the duration of it, because I could do the giveaway now. I could do it in 30 minutes instead. Although as it tends to be, I tend to do it towards the end of the stream. But if I reference something early in the stream that ends up being the key to the giveaway, then only the people who have tuned in earlier will be the ones to get in on that. So, wearing this, usually I have my bug themed shirts, but this was the experience last week, was the gotta catch them all. That's not Pokemon's slogan anymore because there's like literally 1,000 of them, and it's almost impossible to catch them all. But it certainly was what it felt like with all the things that we were fighting in Alabama. uh, so, um, so yeah, where to begin? I don't know if I want to start talking while we have a couple people, or if I want to wait for the numbers to get up. I guess I could just talk about some administrative stuff off the bat. So first and foremost, and I'll probably bring this up again later, I used to have that shirt. I really like this shirt. Hello, Shane, as well. Good to see you again. I really like this shirt. It's all unique artwork for all the Gen 1s. It's not the original. Although it would be nice if it was the original Sugimori artwork, but it's not even like that. I don't even know who did the other artwork, the more modernized artwork for a lot of the Gen 1 stuff. Which is what, at least back in my day, was what they plastered on everything when they were selling Pokemon paraphernalia. And so to see this shirt with it's done in that style, but everything's all original poses and all that stuff. I really like this shirt. So administrative stuff have been blogs. Blogs will be incoming with some pictures and whatnot. I really wish I could have gotten the ball rolling on that sooner. But honestly, just tired from driving and collecting and having to take care of the things that we collected, and then getting back to work on all the things that need to be done day in and day out here, because there's a lot of them. So blogs will be incoming. I'll probably write a batch of them and have them pop up every 2 to 3 days with a lot of just some notes on some things. It's a very interesting trip overall. And yes, Evan, the original 151 and Gen 2, 3, the ones where a lot of them look more like monsters than cute anime sidekicks are kind of my favorites. Again, to reiterate, the ones that look more like actual monsters and less like cute little anime sidekicks and the power of friendship and all of that jazz. Not as huge of a fan. Anyways, going on from that. Administrative stuff. Tomorrow, pending completion of Will's training and blah blah blah, will be Will's last day working at Roach Crossing, and he's been doing contract work. I mean, this is can't remember 1095 or whatever forms that that he has to fill out and do, but he's been on basically full time, or as much as I need him to, for the last, boy, it's already been like 6 months. And so he's my little boy is moving on to a big full time job with all the benefits and whatnot, doing vegetation assays for one of the local companies, also being contracted in for that. And so he will be dearly missed. Will, if you're watching or if you're not watching, you have been a wonderful asset and companion to Roach Crossing. And I hope that the new job goes swimmingly and you don't find yourself hating it or anything like that, and that you can find plenty of cool pseudoscorpions and plants while you're out on the job. So, but unfortunately, what that means for me is until I get a replacement Slav, is I will be at 50% staffing capacity, and that will be, it's not debilitating, but it will certainly cut into the promptness with which I can respond to emails, as the collection has grown, perhaps doubled or maybe even tripled in size in the last 365 days. And I take pride in having invested the initial time and materials into making many of my enclosures relatively self-sufficient for long periods of time. There's only so much time that you can buy with equipping an enclosure. So as good as my setups are, there's still maintenance that needs to be done on things, and if things start to go south, then obviously I have to do some pretty heavy maintenance on stuff. And so it will be a bit of a struggle to find a balance in the next couple of weeks. So I appreciate everyone being patient as the dust sort of settles and I begin looking for a replacement basement Slav. Brandon and Will have both offered to come and do work, odds and ends stuff here and there. So there's that. But if Will's vegetation assay room coming soon. So so that's where we will be at starting on Monday, basically. And I've gotten through about half to 40% to 50% of all the emails that I have missed while out, fortunately. And so if you're one of these people who I have not responded to yet, I am working on it. It may be done after the live stream. It may be done tomorrow, but within the next 3 days, the inbox should be up to date. So availability on the site will probably take a bit longer. I need to go through and see, especially for things like isopods where cultures are colonizing the bins that we put them into, and I will actually be able to switch a lot of things from contact and not available to fully available. Kyle, I'm going to take a moment to respond to some of these comments here. When you go out in the field, do you bring a book and pen with you, or just note everything on your phone? So I usually, if it's something I'm not familiar with, I will sit there and sort of digest the situation in my brain as far as, where something's found, the sex, the numbers, what it's associated with, etc. And that forms a very vivid picture that sticks in my brain that I can think back on very easily. For locations and stuff like that, I will remember, at least for the first day or so after collecting things, basically where I found it, as far as like Dearborn, Michigan, or Ann Arbor, Michigan, or whatever. very bad examples because I would never forget collecting anything in Livonia versus Ann Arbor. But going out to a foreign state, um, I will be I will usually at the end of the day before going to bed. A lot of us, if when we're in a collecting group, we will go back and check on our hauls and look how things are doing and see if anything's eaten anything else. Blah blah blah. And at that time, I will jot down some shorthand on the containers that will remind me of if it's what's inside it in some cases, if it's not readily visible. So like some things like some of the spiders that I collected, I would write down, what spiders in there are shorthand for it, and then the place, something brief to remind me. Not always the place name because, excuse me, I don't always know or remember that. So I'll write something that like we were going to some bowlers loop in Alabama. And so in all the containers that I collected there, I put like loop and then whatever, the contents were. And then I will go back when I'm setting up enclosures and I will pester other people who are with me for GPS coordinates or for the city name or the place name or whatever. And then that information will be logged on there. And then eventually it settles in here. And then eventually it goes onto the website. So, you got mine. Thanks again for the info. So yeah, I hope that sort of answers your questions with stuff. So let's see. I guess we got a comfy number of people, so I guess we can dive into the trip. So the night before the trip, we left on St. Patrick's Day. The night before the trip, I decided to now looking back, unwisely do some drinking because Will and I were not going to be somewhere or in a state of being to be able to enjoy St. Patrick's Day as God intended, and so I actually ended up drinking a little too much of something that I did not think was as strong as it ended up being, and that ended rather unfortunately in the night with respect to my digestive system. So the next morning, off to an amazing start. Not feeling very good at all. Very sleepy. Very alcohol poisoned, etc. But we eventually made it all the way down to Alabama on that day, and then we went out with. We stayed at Alan's apartment, which is in Auburn, and um we we went out late at night to go get some fast food. And even though Auburn is a college town, and even though there were tons of options, we just couldn't find anywhere that was open that had like staff that were attentive. We went to a McDonald's and they were open, but waited in the parking lot for like 25 minutes, and ended up having to go to a Waffle House and getting I just remember looking at the hash browns that I got, and they just looked like somebody just grabbed a handful of them, just slapped them in the container. And so that was our dinner that day, and getting adjusted in Alan's small apartment was fun. Then the next day, it rained. It was heavy rain when we woke up around, I want to say 8 or 9. And so we just sort of stayed in Alan's apartment and talked, and Alan played Brawl Stars, as Alan often does, um, and talked about bugs we could find, things we could do. And then I had been coordinating with Dr. Appel from Auburn, who Alan is doing his work for, to come and talk to them about things they could use for future studies and cultivation stuff, and just sort of a general meet and greet each other on things like that. And Dr. Appel is a very busy person, so when Alan took us to Auburn, he was in a Zoom meeting still that had gone over time, and so Alan took us into the back rooms at Auburn to look at all the cockroach cultures that they maintain, and it's just tons and tons of German roaches and tons of American roaches, and a couple of small colonies of other things, because they do a lot of work with pest species for bait developments and whatnot. And then Dr. Appel's personal interest seems to be having a large cockroach collection that's very easy to maintain. and also being able to occasionally dip into that to use for their comparative physiology and just general usage of all kinds of fascinating 50s style exploratory science. And so I got to go around the back rooms, saw some things that I wanted that I didn't know they had, and saw all the German lines that they have, and saw some things out of there that I wanted and whatnot. And so eventually, we went to meet up with Dr. Pell when his meeting was over. So that was very pleasant. We went out to lunch. I sort of pitched drain flies to Dr. Appel for his research because, as everybody who's been in the stream has seen, a large German cockroach collection seems terrifying to me. Yeah, with that many Germans, there were some escapes, but the building is so dry. It is a death sentence for anything that gets out of those containers that doesn't have the water available to it. And they also, the USDA FIS certified, so they have all the bait traps and stuff around in case other things get out. So it's just part and parcel of doing that sort of work with things. They can't use like the little electrical grid stuff to keep them contained either because of the humidity and whatnot. And even in the home, Germans are not a huge deal at least in homes that are maintained drier. I will begrudgingly admit I have lost a German roach or two, not only into this house but in my parents' house. Nothing has become of it. And this is actually something that Dr. Appel and the group of us talked about was a big part of their research is finding out that to kill roaches, they have to be happy, or at least to actively kill them, they have to be happy. So he was talking about how, they can take different life stages of German roaches and try to run all these tests and stuff on them to see bait effectiveness and moisture preferences and whatnot. And when they're alone or when they're lonely or unhappy, they just sort of go into this just very core survival mode where they don't want to take baits, they don't want to move, they don't want to reproduce, they don't want to grow, they don't want to do anything. And they will basically just sort of exist for a very long period of time and then eventually develop and then die, etc. But so when they do these testings, they need to have happy, active roaches basically in small colony or group settings, because with just one or two or with just one or small numbers, they just don't do things. It must be very hardwired into roaches or into some roaches. This is, I think, a reference to the Germans, but it has applications to other roaches. Is when they're just alone, they just go into preservation mode, most likely in an attempt to not die before they find a partner or a group to reproduce and to bum around with. So yeah, that was very interesting. And he was also talking about how the idea that they won't get comfortable by themselves makes sense to me. I couldn't keep Germans with how humid my area is. Yeah, and it's very easy to kill them. I've killed multiple colonies of Germans either by desiccating them or keeping them too moist. They're surprisingly finicky. And again, going back to the whole numbers thing, is it's better to start with more if you can when you start colonies. And in these situations, it's like if you don't dump 100 German roaches into a house, the chances of them getting established just decreases because they don't have that elite effect, which is something I brought up before. I think maybe in the Roach Care Guide, but multiple times in the blog and a couple of times on the cure pages, is basically, they need to feel comfortable to keep their life cycle going effectively, basically to be efficient. And so when there's very few of them, it's more difficult for them. One of the reasons for the reputation as household pests is, apartment living, condo living, etc. If you live in an apartment complex and somebody has a German roach infestation and doesn't know about it or won't do anything about it, and you end up getting one because the group starts to move into your area and you exterminate it, and all of a sudden they're back, then that's basically why. Is it gives, in some ways, it bolsters the idea that they're indestructible because you're not wiping them out across the board and whatnot. So basically, once they're established, they're very difficult to get rid of in some contexts. But getting them freshly established, you're not going to move into a new household building and suddenly have a cockroach infestation. That takes time and sort of a building up of cockroach culture, if you will, within that space. And with American roaches, the pest status is not that they breed indoors, but because they use sewers and drains and other things. And even if there's no sign of any of an infestation or group within your house, if they've colonized the drains and the network goes all over the neighborhood or all over the city, they can come out from anywhere at any time, and there's nothing you can really do about it. So I've dealt with a German infestation. I'm scarred for life. Yeah, that was moving off of, or continuing on with the whole German roach study. Was I was picking his brain about how they've been able to get different insecticide resistances, and he was talking about how sometimes it can be epigenetic or acquired where they'll become resistant to different things, and sometimes it's genetic. So you wipe out 99% of the population and all of a sudden you have 1% that's resistant to whatever you're using, which is why a lot of pest control companies rotate through different insecticides, is in an attempt to just completely wipe out everything under the guise that, well, you wiped out 99% with pesticide A. You come back 3 months later, you hit them with pesticide B, and then you knock out 99% of that 1%, and that could be functionally all of them. So by rotating, you're covering all your bases and making sure by killing them as cleanly every time as possible that you're not going to leave just enough for them to develop resistances. Um, so that's the logic there. But he was telling me that they can stack resistances to things. There don't seem to be trade-offs to having more and more resistances. There is not a significant fitness cost on the roaches in a captive setting for them to continually build resistances at a population level, which was very interesting to me, because usually when there's those sort of things, there's a trade-off. For, just as a random example, maybe the resistance comes from them making a certain type of protein. Well, if they're producing this protein and other roaches are not producing this protein, there's energy expenditure into producing this in the form of, oh, well, they can't reproduce as much on the same amount of food, etc. So it's interesting that in captivity, that there is not enough pressure from restriction of resources to prevent them from just building immunities to a bunch of different insecticides. So it's not a limiting factor in that. On roaches carrying diseases, anything can be a vector for diseases. With American roaches, it's, if they have anything on their exoskeleton when they crawl out of the sewer, they're going to transmit diseases. They don't seem to be. There's some data on some strains of Paraplaneta americana having, I think it was salmonella in their gut, but it's questionable if they're a functional vector of that to humans. So that's kind of up for debate. It would seem the bigger threat from having a roach infestation boils more down to the fact that many insect proteins, not just roach proteins, but insect proteins in general, are allergens. And so that's more of the health cost associated with roach infestations, is inducing allergies and immune-related things, as opposed to directly being a vector for diseases, as things like rat feces and whatnot are. It took a year of regular pesticides to get rid of them, and it was a solo building. They might have developed resistance now that I think about it. Yep, they're just think about the ability to develop resistance on a population scale like bacteria, but but you know, but it takes longer because they're not bacteria. The roaches, they breed like roaches, but it's not, dividing every 18 hours or whatever the rate is for certain bacteria. So um so yeah, basically they could stack stuff, and they had a bunch of cultures of different lines of Germans that they had collected from places where they developed resistance to different pesticides. And so one of those cultures, there were some very light colored Germans. Actually, this leads into another important thing that Dr. Appel and I had discussed. This leads into another thing. Uh, where was I going with this? Lighter colored Germans, and I actually I wrote down the strain number, and I actually was able to get some of those Germans from this strain. So I have no idea what they're resistant to, because there was just a code number on the culture, which is probably cross referenced with something else in the lab to say, oh, they're resistant to this, or oh, they're this. So I now have a culture of German roaches from Auburn that has some resistance that I don't know what it is. So um, Darian Johnson, can you show some grasshoppers? I actually don't have any grasshoppers hatched out right now, waiting for lubber hatches and then soon rainbows, hopefully after that. So once those have hatched out, then I can start showing them off on here. Chris Schneider, benzoluria chitin synthesis inhibitor, Psychic CS maybe. I will say the pesticide we used was supposed to target chitin. Yeah, so there's all kinds of workarounds that in a genetically diverse population are just waiting there. Either things are het for them, or they end up being homozygous recessive, and you put that selective pressure of the herbicide on there, and you kill them down to whatever's resistant. Then the next generation is going to be completely resistant or nearly completely resistant, depending on the exact mechanism of the resistance. So yeah, it was interesting talking to him about that. I we Another thing, the important thing I was to say was that culture of Germans I got that has the strain label had some reddish individuals in it, and I had brought up to Dr. Appel that I was thinking that reddening that you see in some roach cultures under certain conditions, I had brought up that it could be a Serratia infection, which is something I talked about, I think, last year in a blog post. Um, and Dr. Appel confirmed that yes, it is a Serratia infection that develops in the roaches, and you can't do anything about it. There's probably certain strains of it that are more effective at colonizing the roach cuticle than others. And basically, you could have a healthy colony for years, and there'll be some sort of stress trigger, and all of a sudden you'll have a breakout of individuals that have the Serratia infection in them. And I can confirm that because I still fight battles with it popping up in a couple of my Bursatria cultures, in my Eublabrus CF biolii, the 6-spot roaches. And recently, my Hemiblabra have been getting hit more heavily by the Serratia infections. But I was able to, as of now, it seems completely eliminate it in my Archimandrita tessellata, which is great because that colony was fighting for years to get me the number of individuals it used to be at, so I could actually make them available. And now it seems that I was able to get rid of the Serratia, which seemed to be inhibiting some of that good reproduction. It's a gram-negative bacteria. I'm gonna look this up. It's if you've seen the sort of scum that forms in your bathtub or in moist places in your house or apartment or whatever, that's sort of a pinkish red. That is Serratia marcescens, rod-shaped gram-negative bacteria. And we were talking about treatments for it in roaches, because I thought, well, it would be great if I could just treat the colonies with something that would wipe out the Serratia, and then I would just, it would be gone, and husbandry would be good again, so I wouldn't have any more flare-ups of it. And Dr. Appel told me basically when you treat roaches with antibiotics, because of the slurry of microorganisms inside of them, you usually end up killing the beneficial bacteria in the roaches, and then of course they can't survive. The fitness is just so poor that the colony doesn't recover. So it seems there's going to have to be a holistic approach, and I'm going to do some heavy investigating, maybe even selectively breeding the strain. I really don't like having to. I really don't like having to breed a captive culture to be resistant to something like this because, again, as we've been discussing with genetic diversity, breeding for resistance to something could also be affecting other components of the genetics that would take away from the diversity present in the intact strain. So, anyways, I have to do some more thinking about it and see if there's anything else that can be done that would sort of help in that sort of way. So, anyways, so now with Dr. Appel, I had talked him into working with drain flies, which Alan is not happy with me because my obsession has been drain flies the last couple of weeks, as everybody's seen as I brought them on the stream. I can also say it's been a month since Will and I collected the drain flies from the drain in my basement, and we had the F1. Well, they weren't really F1s; they were the ones collected. No, these are okay. We had the ones that emerged right before the trip were. I gotta think about this. Yeah, okay. So we've had F1s emerge in the big culture container I put everything into because I put them into their final culture enclosure, the clogmias. And now, by the time that we got back from the trip, it's been another week, and there are babies. So we're on to an F2 of the Clogmia CF alba punctata. And so I was able to talk to Dr. Pell and tell him that, hey, you've got 3 species here that you can use for comparative research. One, a household pest. One that is economically or ecologically important species because the Psychota are usually found in sewage purification places. And so there's this, you have a species that's bad, you have a species that's good, and then you have Lepiciodina, which is a wild species that is completely unassociated with the other two. And so it provides a hotbed for doing comparative research on why do these ones colonize these places? Why is the morphology different? Why, there's a bunch of things that you can look into and very easily test. And they're so easy to rear very quickly, as everybody's seen. It's been a month, and I'm up to hundreds of the clogmia. Very easy to rear them. They don't need special temperature. They don't need special diet. They don't need special enclosures. And Alan was just sitting across the table from me, just glaring at me because Dr. Appel might get him to do some work with those for his graduate work. So, Evan G, some drain flies near a rotten tongue twister, rotten stump slurry. I would be interested in acquiring some of those if that stump is still accessible, because I would like to build my colony, my species count of drain flies, including native ones. I'll bring this up as we go further into the stream, but I actually did some ridiculous things to try and catch some wild ones on the trip. Damn, I want to do that research. Well, let me tell you, it's right there for the taking. Maybe shoot Dr. Appel an email and see if he's looking for graduate students, which apparently he is looking for graduate students because he and I discussed things. I can't commit to that because I have Roach Crossing to run and I don't want to move to Alabama or whatever. But we're gonna see what I can do to sort of help the research along and go from there. Yeah, if I can find some after the rain tonight, we'll send you an email. And if you can grab the slurry, that's even better because it's almost certainly there will be some larvae in there. So, moving on from there. So we went up with Dr. Appel. I got to meet one of Alan's co-students, and I can't remember her name, unfortunately. She's doing work with, there is a parasitoid wasp that goes after Supella longipalpa ooths, and she has a bunch of colonies of it being reared in the laboratory there. What's up? But anyways, so and she's looking to see if Victor, what's up? Let's see what he's barking at. Might be a cat in the front yard. Oh, you're right there. Okay. What? What? You'll come and say hi? Want to come and say hi? Huh? Yeah, no? Okay. Well, go tell him. Go tell him. Go tell him. Go tell him. What do you smell? What do you smell? Go tell him. You guys show me something? I'm going to see. He's got his hackles up, so something's irritating him. I'm going to go see what he's barking at. What's up? What's going on? Oh, what's up? What's up? Go outside? No? You want to come say hi? Yeah? What's up? Come on, come say hi. Good boy. What's up? You smell something? There's also a new cat in the neighborhood. I see it right across the street, actually, right now. It's on my neighbor Cindy's porch. I'm surprised that the what's his name, Charlie the orange tabby, who is older than dirt, has not tried to beat the life out of it yet, but maybe they've already met up. So anyway, so met up with that with Alan's co-student who is working with the Supella longipalpa. What's up? You want to lay down? Maybe? Do you just want scratches? You are a little attention whore. Attention whore. Who's working with the Supellas and a parasitoid wasp, two parasitoids actually, I think, that specialize in attacking the ooths. What? I think he sees the cat. Anyways, and so she's doing work with. Every time I see a cat in my yard, I try to chase it and traumatize it so it doesn't come back. Yeah, I don't. Charlie is fine because he's old. He was old when I moved in here, and he's so old that he can really only go after like baby. What's up? That's me. That's me on there. What? That's me. What are you staring? Look at those eyes. What are you staring so intensely for? What's up? Oh, big silly. Charlie's old, and he only goes after rodents. I've seen him go after rats and mice and a lot of baby things. He'll get baby bunnies, and he'll get baby squirrels too. And I'm always like, how did he catch them? Because he just looks like he's falling apart. And so and I've tried to be nice with him. He does not like me at all. I went to pet him once, and the claws came out, and then it got all infected. And I'm just not a cat person. There are some really nice cats out there, but so we respect each other. I say hi to him, and it would behoove him not to go in the backyard where Victor is for obvious reasons. So, anyways. So Alan's lab mate is working with the parasitoids on Supella. What? And she's looking to see if the parasitoids will. What? That's me. That's me. It's me there. What? Just because you don't understand it doesn't mean you should be afraid of it. And so she's looking to see if they'll parasitize bladdids. She's looking to see if they'll go after other ectobiids. And so far, her work, she's found that they're pretty strict about going for Supella. They've gone after a couple of other ectobiids, but they're pretty strictly associated with Supella. And I thought, since Germans don't have, and she's also tried it with Germans, obviously. Germans don't really have any parasitoids that go after their ooths. In fact, it is thought that the whole incubating the ootheca, as well as the like with Germans, where it's not. What? You want to go outside? Go outside? Are you trying to do it? Come on, let's go outside. All right, put a pin in it. Back to the pins, or back to the chair stream. We're gonna take them out. Come on, let's go outside. Come on, come on. Let's go outside. Come on. Come on, Victor. Let's go outside. Come on. Come on. Come on. Come on, good boy. Just gonna sit there boofing at me. Anyways, so it's hypothesized that the live chair reaction. Evan, let me scroll back up and see your question before I get on a spiel here. If I collected the slurry and kept it to see if any hatch out, would I need to do anything other than keep it moist? No, that should be all you need to do if you want to sort of kickstart the larvae that may be in there. You can sprinkle in some fish flakes or push a piece of dog food or a piece of cereal into the slurry for them to eat, and then just basically wait and care for the adults, and the subsequent generation should be the same. So, anyways, with the parasitoid research, it's thought that bringing the egg case into the body cavity and also as an intermediate step, holding off the tip of the abdomen is actually an adaptation to prevent things from parasitizing the egg case, because the thought being that if it's inside the body, things obviously can't get at it to lay eggs on it. And as sort of an intermediary step, we're just sort of not completely committing to carrying it around like that, or for things that hatch out faster, just holding it on the end of the abdomen also gives some of that protection, because the adult roach can feel if there's something touching it or going for it and kick it away or whatever. And so she so that's the work that she's doing. And seeing how effective and how ruthless those Supella parasitoids were made me realize, if they're out and about in places, if it's not just Supella out there existing in the void, and if the parasitoids were brought to the United States or wherever they infested with them, I could very easily see why Supella is not a common pest roach, even though it's very tenacious. If you get them, they don't seem to quite reach pure infestation levels in a lot of cases, but yeah. So Supella is definitely very well controlled by the parasitoid, and so yeah, we did that. Alan was able to get us into the bug collection, the dead stock collection. Will wanted to see their pseudoscorpion collection. I went to look at their roach collection. It was only like three drawers full. There were some neat species in there. There's some ladybug mimics and whatnot, but I'm the livestock kind of guy. So, but Will was having a great time looking at all the pseudoscorpions and seeing that one of the guys who named a lot of species in the U.S. actually contributed specimens to the Auburn collection very shortly before he died. So that was cool. So then after that, the rain had stopped, and it was, I think it may have been just barely drizzling. So we went out collecting a couple of places, found a bunch of millipedes. Where did we go? This is where I have to really rack my brain. There was a lot behind a forestry building where we went, and I was able to collect one of the targets, which was Onomerus. There's a very very tiny, and tiny is putting it lightly. There's a tiny pill millipede that is native to the southeast. There's a couple of species, possibly a couple of genera. It just somebody needs to look at it and re-describe, sort out, range map, blah blah blah. And so Alan found this population of them there, and I've raised them before and I've bred them before, and then lost the colony subsequently. I think it got too humid or too stuffy, basically. So yes, we did. We found Onomerus or CF Onomerus because there's another genus, I guess, that it could be. So, trying to think what else we found there. Was also a spot for Lygidium, which is a terrestrial isopod that's very closely related to more marine taxa, I think. Yeah, it's in the Lygiidae, which is Lygia's in that too. So it's a fully terrestrial isopod that is more closely related to extant marine relatives than other terrestrial isopods are. So we were also finding those there. Restarted my sexy legs cockroach culture because there were some to be found there, and I was able to find quite a few of them. Trying to think what else, and then also trying to think where the heck we went after that, which is where the brain fogginess comes in with the blur of the trip. So we found those. I found some Uriurus or Auturus. I mean, this trip was basically millipede collecting in a lot of ways. We found so many different millipedes wherever we went to, and I was able to get hopefully good starter groups of pretty much everything, and I'm still in the process of putting those away. So that night, we went out and did some collecting in Tuskegee National Forest, and highlights of that: termites, some Hemiscolopendra, a bunch of Parcoblata. Which was one of the things I went down there to get, was to get a bunch of different Parcoblata species that I have inconsistent success with. And so we were collecting, and Lata is usually the most common species, but I'm pretty sure a lot of the stuff we collected there was actually Virginica. So that was fortunate and unfortunate because I don't have a good Virginica colony going. Unfortunate because I really wanted Lata, but it didn't matter. So we went back to your house afterwards. Hello, Alan. Nice of you to join us. Yeah, I don't really have to detail some of the in-betweens. I was just trying to think of where we went after we did the collecting around Auburn. So went back to Alan's afterwards, sort of sorted through some stuff. Oh, rewind to when Will and I were driving down to Alabama. We went to a rest area, and we were just itching to collect something because it wasn't freezing out. I mean, it was like maybe high 50s, so it was a little chilly, but it was not chilly for two seasoned Michiganders. And so we were looking around there. I think Will maybe found some pseudoscorpions, but there was an area in this rest area that was kind of off to the side, away from the main area, that had a bunch of pine trees, big, large diameter pine trees, and they were routing the water away from the rest area using some sort of like a little concrete canal type thing. So it was maybe only a couple of inches of water. And then eventually it became a bit more feral as it got further away from the rest area. So it went from being concrete to being a more natural creek bed. And I say creek, but it was maybe only like 2-3 feet across at its most. And so Will and I went looking around there for some stuff, found some mantle slugs or some Venonies I didn't collect. I don't think I saw any Parcoblata, etc. And so when I looked into the stream, and I looked at the bottom to see, there might be something out, fish, whatever. And I looked, and it was like, the bottom's moving, and I can't really determine what exactly it is. What could be this numerous on the bottom of this little creek? So I got down and looked closer, and it was nighttime, so I had to use a headlamp. And so I get down and I look closer at it, and I realize it's a bunch of aquatic isopods just covering the bottom of this little concrete creek thing. And so it's just like, ooh, this is great. And so, but then trying to collect them was a different story because the concrete was smooth enough that the isopods were not just pluckable off of surface of it, but it was jagged enough that they could sort of tuck themselves against it. And even though there were hundreds or thousands of them, to actually collect a couple of them, I had to like sort of swish them up and try to detach them and swipe my hand. And then some of them got knocked downstream and they're flailing in the water column. And I had to catch them, before they go over the little mini waterfall where it turns from the concrete to the natural stream. And so I collected, I don't know how many of them. It felt like enough to get a colony going from there. And then Will and I decided we were going to finally head to Allen's. And so, and again, this is when we first got to Alabama before we even met up with Alan. And so we started walking back to the car, and Will found a tree that had sort of a split in the center of it. It was still alive, but it had a cavity in the heartwood. It was only a gap, maybe only about that wide, and just teeming with life. There were roaches, there were drain flies, there were cave crickets. I caught as many of the cave crickets that I could, and Paraplaneta fuliginosa roaches, the smoky browns. And then, looking closer into it, and the sludge at the bottom had a bunch of fleas in it, and there were some. We scooped a little bit of it out, and there was a hister beetle, a little teeny tiny hister beetle, and there were some drain flies in it. And even though it was crawling with fleas and flea larvae, I wish that we had collected some of it just to keep somewhere and hopefully fish out the drain flies and the hister beetles that were growing in it, because it was just like pulpy wood, roach poop, rodent poop, etc. But it wasn't dusty. It was all liquidy and gooey, so it wasn't like inhaling it would have given me some sort of severe disease. It would have been very easy to just pick up a glob of it, put it in a container, and then just watch it to see what emerges. Pseudoscorpions do not fluoresce under a black light. We actually found that out on the pseudoscorpion stream that Will and I did with Russ from Aquarimax back in May of last year. We tried a couple of different taxon. They did not fluoresce. So, first time anybody's tried that with that many taxon in the same place at the same time. So that was valuable knowledge. Anyways, I don't know how I got on the goopy tree hole. Just to add to the amount of stuff that Will and I found with the aquatic isopods. So we go back to Alan's apartment. We get ready to leave to go to Alan's parents' house and spend some time collecting around there. And what did we do? Where did we go that morning when we left Auburn? Alan, do you remember what we did the morning we left Auburn? Because I can't really remember where we first stopped to look for stuff or what exactly we did. Because I remember once, or we spent the day out Wilson Elementary School. Oh, why am I having a hard time remembering what we found at the elementary school? Divisa. Okay, so that was the first Divisa spot. I forgot that we went there before we went to your parents' house. That was the morning that we left from Auburn to go to Montgomery. Then was the Divisa spot, right? Which was some woods that are planned to be developed eventually. That place, right, Alan? Was like the woods with the vernal ponds and stuff all through them. Yeah, it's again the blur of everything. No, we went to my house on Friday night. Oh, we got there at night and then in the morning. Okay, thank you for correcting that. Now everything can flow. So we went to Montgomery, spent the night there, woke up and went to the elementary school, parked there, and went across to the woods that had beautiful vernal ponds. It's an area that is slated to be developed eventually, hopefully not in the extremely near future, but if you go 5 miles down the road, there's, they've started clearing it for condos and putting that stuff up. So beautiful vernal ponds, dwarf palmettos, and zephyranthes lilies, and just beautiful eastern United States spring habitat. You got the violets blooming, you got the rue anemone, all that stuff going on. And so we went there looking for. Alan says there's a lot of black Parkablata divisa there, which is interesting. And I wanted to bulk out just having this species divisa, so we went there. And Alan caught most of the Parkablata. I was distracted by the plants and by everything else that was there. Cormu aspersum. Oh, that's the garden snails. Yeah. So this was again. This is relatively intact for us. I mean, there was Japanese honeysuckle everywhere, but all the other plants were native and beautiful spring stuff. And so Alan was collecting the divisa. Will and I were kind of flailing around. I did grab a couple of the dwarf palmettos, and eventually, while we were walking into the woods, a fair ways away from the actual vernal pools and ponds, there was a hole, just a hole in the ground, maybe only a 1.5 feet diameter, just sort of away from all the vernal pools and stuff and whatnot. And Will and I just sort of looked at it, and with our past experience with strange holes in the woods and strange bodies of water in the woods, we just sort of thought, like, maybe there's something cool in here. And so we started poking around, and this hole, which is very clearly fed by groundwater, it's not like a spring or anything like that. It's not actually flowing, but it's more like just a depression that is low enough and in the right place that the groundwater seeps into it. There was a bunch of amphipods and some isopods, and the water was all. I'll put up a picture of it because I took a picture of the whole setting. I'll put it in one of the blogs, but it was silty colored, so it was sort of a smoky light gray tan, and you couldn't see the bottom of it. So I had to put I had to put my hand into there all the way to the bottom if I could, and I didn't commit to going all the way in because south could have been cottonmouths, whatever, hiding in there. So I just sort of went in as far as I felt comfortable with, and it ended up going down into the side, so it's connected to something else, or just went deeper. And so we collected some of these amphipods and isopods. I was primarily after the amphipods because at the time it looked like they had no eyes. So they were just sort of a ghostly white color, and it looked like they had no eyes. But upon closer inspection later, found out they have just the teeniest, tiniest eyes, very teeny tiny on an otherwise creamy white amphipod. And I did end up catching a couple of the isopods that were in that water as well. So that was one of the exciting things from there, and it's kind of been the gift that keeps on giving because they did make it back to my house alive with no noticeable losses. Not just a good number of big ones, but surprisingly a lot of smaller ones that must have been hiding in the leaves that I collected from there. And I set them up in a 5.5, and they've got an aerator with a light bubbling, and they've got some aquatic moss. It's actually not Java moss. It's a locally collected aquatic moss that Will and I have been culturing for a couple of years. And so have this little ecosystem tank. I'm curious if any Daphnia or Moina or copepods are going to pop up in there too. There were also some aquatic segmented worms, some aquatic oligochaetes that fell into there too. And so I'm hopeful. They seem to have adapted to. I was using spring water. I should have went and got a big container of spring water from Meijer or Walmart, but I ended up going to Costco and buying the bottles of water. And the environmentalist in me was screaming as I was dumping the spring water and bottle by bottle into the amphipod tank. So next time I'll just go get a big one or do some aged water and go from there. So anyway, so that was one of the big finds for us from there. When some egg says, when you collect aquatic invertebrates, I assume you don't actually transport them in water, or do you? I do transport them in water, and it has a good success rate for me. The things that I found is you can't let it get too hot. You have to, and if it does, if there's a chance of it getting hot, you have to make sure there's some sort of ventilation. Otherwise, it'll just steam up and boil. And anything that might go to the surface to try to get air. Well, I think he wants to. Um, um, Anything that wants to come to the surface to get air or whatever, if the water gets too warm, if the water, if the air column is hot or too humid or whatever, they'll just suffocate and die. Um, so, and what was another thing? To make sure you get just the right ratio of substrate or stuff for them to eat or hold on to actual fresh water from the habitat. Um, and so and I walk the fine line, and it's always like this could just fail, which is what I thought would happen with a lot of the aquatic things on this trip. And it was surprising because out of the things that we ended up collecting, the one thing that was a complete failure was the glass shrimp. Those just all died, and I think it was because I didn't give them enough ventilation and blah blah blah. But all the amphipods and all the isopods that I had collected actually did very well and made it back in good shape. So so yeah, that's sort of the technique that I use, is making sure there's enough of the water collected from where they were. First got into exotics, it was with aquariums. Since then, it's branched into reptiles and inverts and so on. But I've always loved the idea of a no fish or invert only aquarium. What about minnows? Oh, I'll get to the minnows in a second. Sounds like Victor wants to come in. All right, no, he did not want to come in. So here I am, back again. Um, let's see. What about minnows? So also, and I'll get to this later, but I collected some killifish for a friend, and those also made it back without any problems too, and in excellent shape actually too. They showed no signs of stress basically. But continuing on with the inverts thing, I do have a couple of. I do have most of my aquariums downstairs are actually just invertebrates. So we have the Montgomery forest hole tank, which has the copepods, the oligochaetes, and the sparing number of isopods that made it into there. We have the Auburn ditch isopod tank, which is doing very well too. And then in the works is also from another spot we went to a vernal pond aquatic isopod, amphipod, and Alan. I think there were actually some of those freshwater limpets in there too. So that's another tank that I'll have. And then I have some shrimp, and I'll be working with hopefully with sponges in a couple of months. And I would like to give the glass shrimp another go, because I think the big problem people run into raising those is not that they're difficult, but because I think the larvae, they have a larval stage and that they need a lot of green water suspended things that aren't zooplankton. And so I have some green water cultures going. So I would be giving my hand at raising glass shrimp in captivity in large numbers. So I think it's a lot of fun. It's kind of like with the feeders for reptiles and stuff. You start off keeping the fish or the reptile, and then you end up liking the feeders for them more, and you find it more rewarding. So There is. I'm keeping it a bit on the down low, but there is a very exciting aquatic isopod in California that I have been waiting to get my hands on again. And the person who collects them has to wait for the right time of year so that the place is accessible, because otherwise it's just off in the middle of nowhere with a bunch of stinging nettle and stuff. So, but hopefully those will be coming my way within the next couple months, and then if I figure out the husbandry, they'll be made available. But I think that there's something that a lot of people get excited about because of how active they are and what they look like. But I'll touch on that more when I have them in my hands. So um, let's see. Really cool to have an aquarium. I really want those freshwater polychaete worms. It's a weird obsession of mine to try and get those. Getting things that are primarily marine taxa, getting the freshwater versions is something I would really like to do, which is also why I would like to work with the sponges. So shrimp are inverts. Where did you find killifish? Killifish are very common in a lot of the United States, actually, at least the eastern U.S. They were in a roadside pond, lake, backwater area, and there's a lot of different species, actually. There are invasive signal crayfish around here. I may catch and keep. Space is the issue. Uh-oh. I think the stream got disrupted. Can anyone see if I'm still moving? Not saying we had any disruptions, but my feed just sort of stopped for a second. So if anybody in the chat can see, am I still audio good, video bad? Yeah, that seems to be the case. I can hear but not see. I wonder what happened with that. Let's see if I can disable something. Don't think so. Thank you for everybody's immediate responses. Let's try Googling this. Video frozen. See what it does. How to fix frozen YouTube video. How to unfreeze YouTube video. It's just weird that the audio is. Thank you, everybody. Probably the movie. Send feedback. Did it do something? I'm gonna see what live stream YouTube live stream video feed frozen. How do I fix it? Maybe the bandwidth? Clear the browser's cache? Jordanella, Florida is, I would like to work with localities of those one day. We'll say this is a marvelous still image. I'm wondering, you don't have access to this feature, you don't have access to this feature, then what do I have access to? Let's see if this will do anything. Feed. Can I type anything? No, this is weird. And I'm trying to think, just refresh. Okay. Thank you for simple leave page. Let's